Okay, so this is going to be eye-opening for some of you. We're going to talk about lactate, and I'm going to try and address a few of the myths that are out there about lactate. We're going to kind of spoil the movie right now. Lactate is not a bad thing. Lactate does not cause you to be sore, doesn't cause you to get tired. But I'm pretty sure that every one of you that is in this class has heard that. So let's talk about lactate. So here are the 4 things that we're going to talk about. We're going to start off with pyruvate, though. Okay, so normally at the end of glycolysis, we form pyruvate. But there are conditions where the amount of oxygen that we have doesn't meet what is needed by the electron transport chain. Okay, and so what happens is we don't have enough enzymes, mitochondria, whatever it is, to take advantage of what's being produced by glycolysis. Okay, and specifically NADH. And so what happens is we're going to have the formation of lactate. And we'll get into that in just a minute. Now, ultimately, one of the things that helps is we have to talk about different kinds of enzymes in muscle fibers. And so fast twitch muscle fibers have lactate dehydrogenase, which helps form lactate. Okay, so a little bit about lactate and lactate biochemistry. As we discussed previously, one of the products of glycolysis is NADH. Well, we need to break that NADH back down into NAD, otherwise glycolysis will stop, or at least slow down. Okay, and this happens in the electron transport chain. But in order for the electron transport chain to function, there needs to be oxygen, right? Oxygen holds onto those electrons. So what normally happens when there's oxygen present is oxygen in the mitochondria takes hydrogen ions, forms water, and so that NADH becomes NAD. Now, whoops, hit the wrong button. If we don't have oxygen available to take a hydrogen ion from NADH, well, pyruvate will then take that NADH, and when that happens, pyruvate becomes lactate. Okay, so if we've got oxygen, the electron transport chain happens. And one of the things that happens in the electron transport chain is, and I just want to reiterate this here, is that NADH turns into NAD because oxygen takes a hydrogen from NADH, and now we've got from NADH, and now we have NAD. Okay. But if it doesn't happen, we still need to take a hydrogen ion away from NADH. But when there's not oxygen doing it, pyruvate steps up and does it. But pyruvate becomes lactate. So the textbook says this. It says we produce lactic acid. It even kind of addresses this a little bit, but not really. So the question is, don't we make lactic acid when we exercise? And here is one of my little soapboxes these days, and I make a big deal about it. I used to actually have last semester I had an entire lecture on lactate. I'm not quite going that far this time. But Dr. Robergs, who works at the University of New Mexico, I almost worked with him for my PhD, says in a paper in 2004, the belief that lactate production releases a proton and causes acidosis, also known as lactic acidosis, is a construct, and as such needs to be corrected. So in other words, what you've been taught or what you've heard is likely incorrect. Lactic acid can't actually be formed in a human being. Okay, lactate doesn't cause the acidity, doesn't cause many of the things that you've been told. So it's actually a big controversy, but based on the science, lactate is a good thing. Lactic acid doesn't really occur in humans. Okay, so, well, why is this? How did this come to be? Why do people think this? Well, lactate formation is correlated with hydrogen ion production. But if you've ever taken a stats class, you've heard the phrase, correlation does not imply causation. Okay, so what this basically tells us that is as we have more lactate, if we measure hydrogen ion at the same time, well, hydrogen ion is increasing. But that doesn't mean that one's causing the other. It just means that there's this relationship. We don't know what's causing that relationship. Okay, it's important to know lactate formation does not cause hydrogen ion production. 20% of the hydrogen comes from glycolysis. Okay, hydrogen ion actually gets used when we create lactate. 80% of hydrogen ion comes from what's being used when we break down ATP. Okay, so there's also this concept of mitochondrial ATP recycling, meaning and it says that it's hydrogen ion balance. What we mean by that is that for everything we use, we take away as well, right? So we don't increase the hydrogen ion, we don't decrease the hydrogen ion. So what does lactate actually do with hydrogen ion? It actually reduces the concentration of hydrogen ion inside the cell. Lactate is a buffer. And so as such, what it does is it grabs onto it. It holds onto it, okay? Reduces the acidity. Okay, we talked about buffers in 220. And so this is preventing large swings of pH inside the cell. Okay, we don't want our cell to be acidic. That's not great. And so lactate helps take care of that. Okay, the muscle cell has a symport, and what that means is it's a transporter. It's a channel, but in order for the channel to work, you have to have both parts or all the parts. And in this case, we need to have lactate and hydrogen ion. Okay, so if I've got a hydrogen ion, if a hydrogen ion leaves, a lactate molecule leaves. And so what this ultimately does is this symporter helps get rid of acidity inside a muscle cell. As a result, we have this correlation between lactate and hydrogen ion. Okay, it's like two keys. So like I talked earlier, I think in one of the videos, it's like a deposit box, safety deposit box. I put 2 keys in, I can open the box. But if I only put 1 key in, it doesn't work. If I only put the other key in, it doesn't work. So in this case, the keys are hydrogen and lactate. If I've got a hydrogen ion and a lactate ion, they'll both be able to leave the cell. And so as a result, I have a correlation between the amount of hydrogen and the amount of lactate that are outside of the cell. So what happens to lactate? So when does lactate production exceed removal? In other words, when are we producing more than we can kind of handle? All right, and so this happens when we have low oxygen delivery or lower than what we actually need. Okay, so as a result, we increase in glycolysis. And this is where we get into those fast twitch fibers that are anaerobic. And as a result, we produce more lactate. And because we're producing more lactate, we can't get rid of lactate as quickly just because there's so much. We're inundating the system. Okay, there's a concept that we'll talk about later in the semester called lactate threshold, or onset of blood lactate accumulation. All right, so lactate is ultimately a good thing because it helps convert that NADA into NADH. So we need this lactate in order for glycolysis to be as effective as possible. It consumes this hydrogen ion that's produced by glycolysis. Remember, it's a buffer. It buffers acid. It doesn't produce acid. A buffer is actually going to reduce acidity. An acid's going to increase acidity. Okay? And so, like we said a minute ago, it's going to reduce the acidity inside the muscle cell. Okay? Lactate actually helps us get rid of hydrogen ion from the active muscle. Another reason why lactate is great is that we can use it. We can use it for energy. Okay, we can take lactate and we can put it into the oxidative pathways, such as the electron transport chain. Okay, we can create glucose from it through something called the Cori cycle. Okay, so the Cori cycle is shown here. So we've got 2 lactate that are being released by the muscle cell. They travel through the bloodstream into the liver. In the liver, that lactate's converted into pyruvate, which is then converted into glucose. Now, it does take some energy here, but we're actually able to create glucose, which then we can use to generate more ATP. So we lose some energy here, but it's possible. Okay, and so this would be considered gluconeogenesis. We're forming glucose from something that isn't glucose. And so that's all that I'm going to talk about lactate for now, but we will get into it later this semester